So our final talk of the session will be from Jonghyun Bae, who is a postdoctoral fellow at the National Institute on Aging. He will be presenting his work on reproducible and fast myelin water fraction imaging using the accelerated BMC-mcDESPOT method.
Okay, so let me share the screen and go into.
Perfect.
All right, awesome. So hello everyone, I'm back again. So this morning I talked about the validation of our myelin water fraction mapping with BMC-mcDESPOT using transcriptomics. So in this talk, I'd like to put my engineering hat on and share our work on technical development of accelerating this method to achieve myelin water fraction mapping within clinically feasible time. Because we can develop all the methods that we want, but at the end of the day, we care about the translated to the clinical setting so that it can be useful to help patients. So this plot shown here to the right demonstrates different physiological changes such as amyloid plaque accumulation, tau pathology, brain atrophy, and cognitive decline along aging, and potentially during the disease progression as well. However, many previous research studies have suggested different microstructural changes that can be found along this process. And it is suggested that perhaps these changes may precede the hallmarks of AD and the clinical onsets. Therefore, detecting these changes in brain microstructures with reliable and reproducible method is very important, as they can be used as an in vivo biomarker to monitor the aging process and the progression to AD. Among these changes, our lab is interested in cerebral myelination. I shared the same slide this morning, but to reiterate for those of you who missed it, our relaxometry-based technique called Bayesian multicomponent driven despot, or BMC-mcDESPOT for short, aims to measure the water content in myelin. And this multi-component relaxometry technique is well established. I think we all heard this over and over again today, but we assume that the T2 relaxation rate of the water trapped inside the myelin is distinctively shorter than that of the intra or extracellular water. We can then compute the fraction of the water in myelin that is contributing to the overall signal and represented as the myelin water fraction. Now, the BMC-mcDESPOT consists of two imaging sequences, spot gradient echo and balanced steady-state free precession sequences with different RF increments. Without going into too deep in the mathematical formulation here, but we can see different NMR properties are being estimated here, such as myelin water fraction, T2 and T1 of the myelin, and extracellular water and off resonance. As you can see from this formulation down here, this up here with the matrix formation, this is high dimensional estimation problem to solve. And to tackle this, we developed the Bayesian approach to that allowed us to measure myelin water fraction T1 and T2 of the two components. Well, the myelin water fraction estimation from BMC-mcDESPOT is great. It's shown its effectiveness in normal aging as well as also in AD patients. However, the fully sampled BMC-mcDESPOT takes 17 minutes in total, which is considerably long for elderly patients to stay still in the magnet, and certainly too long to be implemented in the clinical routines. Recently, our lab presented this work down here at the last year's ISMRM that by utilizing the acceleration in K-space with compressed sensing, we can shorten the BMC-mcDESPOT protocol to 9 minutes without compromising the accuracy in myelin water fraction estimation. However, the accuracy and the reproducibility of our measurement has not been tested just yet. So in this study, we propose to examine the feasibility of further accelerating our method by undersampling in the data space. So this drastic acceleration allows the acquisition time of 6 minutes, and this would potentially provide opportunities for myelin water fraction mapping to be implemented in clinical scans to examine myelin health in aging populations and in patients susceptible for AD onsets, for example. We assessed the accuracy and reproducibility in myelin water fraction estimation with accelerated acquisition to demonstrate the feasibility. So here are different undersampling patterns that we considered in this study. The first pattern here is to undersample the K-space with compressed sensing, and we denote it as CS-BMC. And note that both Spoiler echo and BSSFP scans were undersampled with a CS factor of 2. Next, we retrospectively undersampled the data space in one of the BSSFP images with RF increment of 0 by only selecting 2 flip angles out of 10 that were originally required. And we denoted this as DU-BMC. Finally, combining these two undersampling patterns, we aim to impose more acceleration in the acquisition. We denote this as aBMC. So for the data set, we acquired the data set from a single subject, where we first acquired fully sampled data, followed by the K-space undersampled data for 9 data sets each. And for the accuracy assessment, we averaged the estimates obtained from data sets that were fully sampled in both domains and considered it as a true reference. And absolute errors were computed for different undersampling patterns against the true reference. For the reproducibility assessment, we measured the standard deviations of estimated parameters across 9 data sets in fully sampled reference and with different undersampling patterns. But first, to perform the undersampling in the data domain, we must choose 2 flip angles out of 10 in the fully sampled case. So we tested the optimal selection of flip angles by computing the error of T2 and B0 maps between those from reference, which were fully sampled, and those from different combination of FAs. As shown in this ugly error plot here, we found that selecting flip angles of 2 and 50 degrees resulted in the least error. So we adopted this selection for the undersampling pattern in data space. So here are the results. The first grayscale images show the anatomical references for different slices in each column. And then the average T2 and B0 maps across 9 data sets from different undersampling along with the reference are displayed here. And we can see that both T2 and B0 mapping in different undersampling patterns shows visually identical estimates. And the error maps computed between the estimates from fully sampled and those from different undersampling showed negligible bias. So here are the estimated myelin water fraction maps using fully sampled reference and different undersampling patterns. We can see that the myelin water fraction estimates show higher myelination in the white matter compared to the cortical gray matter. And once again, with visual inspection, we again see very little difference between the myelin water fraction maps from fully sampled reference data and those from the undersampled data. We also looked at the different white matter regions, such as whole white matter, body of corpus callosum, splenium of corpus callosum, corona radiata, etc. And as shown in the bar plot here, the regional errors were also comparably small, regardless of the undersampling pattern. Therefore, we concluded that the acceleration in either domain or both domains does not really impact the accuracy in estimating myelin water fraction. Lastly, we examined the reproducibility of our methods. The maps of the standard deviation across 9 data sets shown here exhibit quite similar level, suggesting that by accelerating in either Fourier or data or in both domains does not impact the reproducibility of our methods. More quantitatively, the regional variations of the standard deviations were also reasonably acceptable considering the benefits of the accelerated scans. So therefore, we concluded that our proposed undersampling pattern is robust and reproducible. So in summary, we propose an accelerated BMC-mcDESPOT methods, which achieves myelin water fraction mapping within 6 minutes. our results suggest that our aBMC-mcDESPOT method demonstrates high accuracy and high reproducibility. And this reduction in scan time will hopefully allow this technique to be implemented in clinical scans, which would allow further investigation of the cerebral myelination in aging and in neurodegenerative disease. So I'd like to thank all my colleagues for their guidance and assistance, and thank you for your attention. And if you are interested in applying our myelin imaging method to your study cohort or your study, please reach out to me. We can try to arrange our collaborations. Thank you.
Great. Thank you so much. Right on time. Any questions, Aurélie?
There is one from Olivier. He's asking, do you think the accuracy will be similar in pathological tissue?
That's a good question. Unfortunately, we haven't had a chance to look into any patients' scans just yet, but that's definitely one aspect that we want to look into, make sure that our proposed methods are applicable to patients.
Great, thank you.
I might have one question in relationship to some of the other myelin water fraction maps we saw earlier, where the basal ganglia structures were much brighter. I don't remember them jumping out as much in your images. Could you speak to why your method is maybe immune to this?
That's an interesting point. Well, honestly, I don't have a clear answer as of right now. But we never really saw those issues from our myelin water maps. So yeah, yeah, I guess yeah, we'll have to look into those. It's quite issues that I'm not quite aware of, so I don't have a clear answer to it just yet.
Okay, thanks. And I've been doing a lot of sort of B1 plus correction. Have you looked at including this into your models as well?
Right. So that's a very good point. Obviously, for the B1 correction is needed. But again, that comes at expense to the, obviously the increased scan time. So in this feasibility study, we didn't include those, but that's definitely in the pipeline along the way for the future developments.
Awesome. Thank you.